acquisition, or whether there are differences that suggest that they should be treated differently. So one of the things in this meeting that we had is that we all undertook to have very specific follow-on actions from the various organizations and participants that were presented. And this webinar is actually to follow up on where we are with those actions and also probably plan for our next steps. We apologize, and I know we do not have all the participants from the meeting, but the idea of having a follow-on webinar, I think, is really, for me, a very good thing to do. We have about 50% of the participants and probably slightly more than 50% of the people who were at the initial meeting present with us today, which I think is a very good thing, and it just shows that we are very dedicated to following up on this issue. So for today, we are really going to have two presentations. The first one will be on follow-up actions and will be done by my colleague, Dr. Petrus Stein, who is a scientist here, a research scientist in the department, and has really been leading our work on DMPA and HIV acquisition risk. And he will look at what the reports, at least what he has, on what actions various organizations and people have taken that they undertook to do at that meeting. And at the end of the meeting, you will have an opportunity, in case you have had some actions and which are not reported, or you have an intervention, we'll be able to unmute your microphone and you can give your report, or in case there are any questions. This will be followed by a technical report on really synthesizing the data on similarities and differences between DMPA and NET10. This was a key follow-up action that was required for WHO to take, and we'll hear this presentation from Renee Heffron. As I said, at the end of the presentation, then we'll have time for discussion and to discuss the next steps, and also for people to provide any interventions or any updates that will not be covered during the presentation. So it's my pleasure to welcome Dr. Petrus Stein to give his presentation. Welcome.
Good. Thank you very much, James, and welcome to everybody once again to this follow-up webinar. What we have done is I've followed up with most of the people on the action plans. So I have collated a table and I've put together some slides with some of the feedback. Not everything has been captured, so the idea would be then to, just after this meeting, update this in a table and to make it available on the SharePoint. Just for your information also, this webinar will be recorded, and there will be a link available which I will send out in a mail afterwards. So if we can go to the next slide, please. And the next slide, please. Just a reminder about on the objectives of the meeting, and that was to review the knowledge of differences and similarities in association with HIV infection risk between DMPA-IM, DMPA subcutaneous, and NET-10, and to identify research, normative, and program actions that were required in the near and medium term, which was really for the next 3 years. So at the end, as James mentioned, we came up with 4 broad action points. First of all, there's aggregation of the progesterone-only injectables. then the counseling and medical eligibility interpretations of the WHO, then follow up on scenario planning for the ECHO trial, and then some specific follow up with the group. Next slide, please. So first of all, to disaggregation of progesterone-only injectables. And the question here was, how do we disaggregate the WHO medical eligibility criteria? And the real issue here is that in future, we should explore disaggregating the methods based on the mode of delivery. So there were quite a few manuscripts that was in preparation or quite advanced at the August meeting. And in follow-up on the review of the epidemiologic, pharmacologic, biological data, in one package, there were 3 ongoing manuscripts, of which 2 were published. First of all, the one from Hapgood and et al. On hormonal contraception and HIV-1 acquisition, the biological mechanisms, which was published in Endocrine Reviews in 2017. I have the link there, and as I mentioned, these will all be available. So the recent paper of Hapgood is not about new research in women. It summarized the existing laboratory and animal data on the most commonly used progestins and links this to existing data on studies of women. And it makes a really useful contribution in compiling these data. But once again, it's not definitive. The second manuscript that was published was Chelsea Polis et al. And that is a lower dose subcutaneous contraceptive injectable containing depot-medroxyprogesterone acetate likely to impact women's risk of HIV and that was published in HIV. Unfortunately, Chelsea cannot attend this meeting, and she apologized for that. But once again, this is an article specifically on the low-dose subcutaneous contraceptive injectable. The article notes that the question remains about how biological responses to DMPA IM and DMPA sub-Q and their potential impact on HIV risk might differ. And the authors write that there's a direct parallel comparison of DMPA IM and DMPA sub-Q that's critical because of the ECHO is not specifically assessing DMPA sub-Q. We will have some update on the ECHO trial just now. Then if we go then to the next slide, there were also some specific follow-ups from FHI 360 who will explore analysis of samples obtained in their studies and to maintain and to maybe compare with the samples from the ECHO study. There are still some, they are still applying for funding, and there's some funding pending for that. The second point was the tissue concentrations of medroxyprogesterone acetate and DMPA sub-Q and DMPA IM, and that was by Sharon Achilles, who cannot attend this meeting either. And unfortunately, there was a vaginal contaminant with a peak at the same place as DMPA that is obscuring the ability to get the accurate MPA concentrations, and they are working very hard on that to see whether there's a solution for that. Then the good news is in a study on the analysis of biological changes comparing DMPA IM and NET-N on day 30, they were busy with the final data analysis for DMPA IM versus NET-N for immune cell changes, and that's not available yet, but they are working in the final stages for that. So the next slide, please. Counseling and MEC interpretation. Here it was USID to continue work on HC3 strategic communication framework on hormonal contraception and HIV, and exploring the possibility of having this implemented in Tanzania. As you saw in the report that we sent, we added the link there to the strategy, and they are busy. They have plans to implement and evaluate the hormonal contraception HIV counseling pilot in Tanzania. I've seen that Saad is on, and maybe he wants to say something just after we've completed the slide set. Then WHO is working with 5 countries in South Africa, Zimbabwe, Swaziland, Kenya, and Uganda to strengthen national actions following the adoption of the 5th edition of the medical eligibility criteria of WHO. So once again here, the good news is that the Global Handbook for Family Planning will be released on the 15th of February 2018 in a meeting in India, and we will have the link on that. And then also we have a follow-up webinar tomorrow with the countries and WHO officers, and I know some of you will be on that meeting as well, on how we are going forward with the specific recommendations and messages in the Global Handbook. Next slide, please. Then scenario planning for the ECHO trial. So the first bullet point there is follow up on modeling application of various possible results of the ECHO trial. So the modelers presented this at a meeting in October, and they are in the process of preparing the manuscripts. It has, we will definitely send you the link as soon as we get that publications. Then the second bullet about the Bill and Melinda Gates Foundation and USID have started scenario planning for the ECHO outcome in collaboration with the Swedish CEDA. And Kim Sheffer is also on the webinar, but the few points that she mentioned here is that in October 2017, USAID, Bill and Melinda Gates Foundation, and the ECHO Trial Management Committee met to better understand the communication needs, timeline, and scenario planning for potential trial outcomes of the ECHO trial. The meeting provided a start to a partner discussion regarding the trial and the issues around various scenarios. So the follow-up plan there is that in February 2018, a meeting is being planned for the ECHO donors and other global partners to expand on these initial discussions. Donors will meet to share what progress has been made to date and address the needs address what needs are outstanding. We expect that more detailed scenario and communication planning and development will take place at that meeting. So the last bullet point on the slide, the East London site ECHO, is to follow up with the potential funders of interest for the witch trial. If you remember the trial that was presented there. I also saw that Justice and Jenny is on the line, are both on the line, and just a follow up that I have is that there's no immediate funding. There's an application for the UK MRC Global Trials Grant, and the outcome will only be in June 2018. In the meantime, they plan a smaller randomized controlled trial comparing hormonal, immunological, psychological, and behavior effects which may impact on HIV acquisition between DMPA and NET-N. Okay, next slide, please. This was just last group for follow-up with the group. As you recall, we were asking if there are any people missing, and we are happy that the suggestion there to include PAS, we invited them, we sent the meeting report to them, and I saw that they were on the meeting, so very welcome to them as well. Then the last bullet point was just the update of the SharePoint. The SharePoint has been updated and it's up and running. However, the function that we were supposed to have on sending out automatic messages that it will, when something new comes up, unfortunately, it's been some problems with, and I think in future we will just send out something that we will add the material to that. So that is it for me at this stage. I am not sure whether somebody wants to say something at this stage. If you can raise your hand, or James, over to James rather. Shall we finish the meeting?
Yes, yeah. So thank you, Petrus, very much for that very nice presentation. I think it's very good to see that there's actually been a lot of follow-up actions following that meeting. For us in WHO, we always feel very glad when the meeting does not just result in another meeting, but actually results in concrete actions, as has been reported by Petrus. I think we just move on to the next presentation, and then we can have all the interventions at the end in terms of people maybe who have additional reports or people who have other suggestions in terms of the way we need to move next. So our next presenter is Dr. Heffron. Renee Heffron is a member of Faculty of Global Health and Epidemiology from the International Clinical Research Center at the University of Washington. And I think it's very fitting for Renee to be the one to do this review, because I think her paper some years ago, for those who know the history of DMPA and risk of HIV acquisition, really kicked a lot of the discussions that are still going on. So welcome, Renee.
Thank you so much, James. I hope that my volume is okay. You can go to the next slide, please. So what my slides are going to show a snapshot of a report that I've been working on in conjunction after reading through the slides and reading many of the references that were presented at the August meeting on DMPA sub-Q and IM as well as NET-N. And so the report is titled A Synthesis of Available Data on Epidemiologic, Biological, and Pharmacologic Differences between intramuscular DMPA and NET-N injectable progestin-only contraceptives. You can go to the next slide. So the report has the standard beginning, and then has a section with acknowledgements and the executive summary and an introduction, and has sections so far laid out for epidemiologic data for DMPA IM and NET-N, as well as biologic data and pharmacologic data, and then a separate section on DMPA sub-Q, because we don't know quite as much about that yet, and then a section that lays out research gaps. You can go on to the next slide, please. The information in the report thus far is based primarily on the slides that were presented at the meeting. And so I want to make sure to acknowledge the presenters from that meeting whose data focus on potential differences between DMPA of both kinds and NET-N, including Chelsea Polis, Janet Hapgood, Sharon Achilles, Lenina Dorflinger, and Petrus Stein, think most of you were able to make this webinar today. So next slide, please. I want to just be sure to note that these slides summarize the information that's in the report, but the report is still in draft form, and it's very timely that we're able to come together now because I would love to have your comments, especially if you can point out places where you think information needs to be added or to be clarified. And of course, these are slides that are giving the information in the report, so the slides are even more brief than the actual report. But I think they'll give you a good idea and you'll be able to give some important feedback. Next slide, please. So in the executive summary, I'm waiting for the next slide to come up. In the executive summary, there's two key points. The first point, after summarizing the data, is that there are important differences between intramuscular DMPA and NET-N in the data that are estimate epidemiologic associations of each with HIV risk, as well as the composition of the progestins. And there may be differences in the way that they interact with potential biologic mechanisms and actual HIV susceptibility. The second point is about subcutaneous DMPA, and that it follows a similar PK trajectory to intramuscular DMPA, but there are few data assessing HIV risk with subQ DMPA, and the biological mechanism that may be elicited by subQ DMPA, there are also few data on biological mechanisms that may be elicited by DMPA subQ to increase a woman's susceptibility to HIV infection. So that's the executive summary synopsis thus far. Next slide, please. The introduction of the report just briefly summarizes that the conversation of hormonal contraceptive methods and HIV risk is very importantly that it's framed through a reproductive rights lens. Also, it describes that there's been a long-standing dilemma regarding the quality and the interpretation of observational data addressing hormonal contraception and HIV risk, and in particular the risk due to injectable contraceptives and intramuscular DMPA. And the third key point of the introduction is that there's a recognized need to determine whether data are currently sufficient to warrant a disaggregation of progestogen-only injectables for the MEC. Next slide, please. So the next parts of the report go on to summarize what we know about DMPA-IM and NET-N side by side. And there's epidemiologic data, there's a summary of biologic data, and then there's a summary of pharmacokinetic data and pharmacodynamics data. So in terms of epidemiologic data, the report includes these two forest plots from recent systematic reviews that look at HIV risk associated with DMPA-IM and NET-N and HIV acquisition. You can go to the next slide, please. And the report also shows the results from recent meta-analyses that included DMPA-IM estimates disaggregated from estimates of NET-N, and the results from the meta-analyses are shown in this table. These are all things that were shown in the slides that Chelsea Polis presented during the August meeting. The meta-analyses demonstrate a point estimate of 1.4 to 1.5 for the association of DMPA-IM and HIV acquisition, and no association of NET-N and HIV risk, but there are limited data on NET-N and HIV risk. Next slide, please. And so as a summary of the epidemiologic data, the main points are that there's potentially a different association being estimated between NET-N and HIV risk and DMPA-IM and HIV risk in clinical observational studies. DMPA-IM appears associated with a moderate level of risk that's statistically significant in observational studies that have the fewest methodological limitations. And NET-N does not appear to confer a statistically significant risk, although data are few and the estimates from individual studies are somewhat imprecise. So that's the summary, or the main highlights from the summary of the epidemiologic data that are so far in the draft report. You can go to the next slide, please. The report now turns to summarizing the biologic data. The slide is a little bit busy, but it summarizes the data that were presented during the August meeting into 4 different sections, talking about architecture, cellular activation, adaptive and innate immunity, and then microbiota. In terms of architecture, a summary of the data are that epithelial permeability appears altered after DMPA-IM initiation, but there are no data about this related to NET-N. A summary of the data on cellular activation are that the data are inconsistent, but there are data from separate studies that show each of these injectables may increase CCR5 positive cells. A couple points summarizing about adaptive and innate immunity. The first is that DMPA-IM has been studied frequently with inconsistent results about its effects on up or down regulation of various cytokines and immune markers. And then related to NET-N, there's been one cross-sectional study that evaluated NET-N that demonstrated increases in pro-inflammatory cytokines. And then in terms of microbiota, the data are inconsistent across studies, but there is the potential to conclude that neither DMPA-IM nor NET-N influence microbiota to a clinically meaningful extent. The data are limited, especially for NET-N though. Okay, you can go to the next slide, please. And so after describing those different types of biologic data, a summary of the biologic data is that there are inconsistent results with regards to identifying definitive changes that are elicited by the initiation of both DMPA-IM and NET-N. The majority of studies have focused to date on IM DMPA, and one recent study incorporated NET-N. And then a third summary point is that DMPA-IM alters markers of multiple immunologic functions, including direct changes to the immune function and pathways that lead to inflammation. Potentially, these pathways are through microbiota and other changes. So next slide, please. Following the look and the summary of the biologic data are some summaries of the pharmacokinetic and the pharmacodynamic data. So on slide 12 or on slide 24, I show the 2 graphs that have been for a long time about NET-N serum trajectories after injection and DMPA-IM trajectories in serum after injection, and summarize those by saying that MPA and NET reach similar peak levels after injection, but that NET declines faster. Next slide, please. So moving on to data from pharmacodynamic studies, some summary points are that NET-N may be less hypoestrogenic than DMPA-IM, but the losses in bone mineral density are slower with NET-N use than with DMPA, and both are reversible. The next point is that MPA is the only synthetic progestin that has a high affinity for binding to the glucocorticoid receptor, as well as other receptors. This type of activity can stimulate and inhibit transcription of target and can be observed through changes in cytokine and chemokine levels. So that may relate back to some of the markers, immunologic markers we're seeing. And then the final point is that NET has a high affinity for binding to progesterone and androgen receptors. And so this draws out a difference between NET and DMPA-IM. Next slide, please. In terms of pharmacodynamics data, this table comes out quite small on the slide, but it was a table that was shown by, I believe by Janet Hapgood during the August meeting. And it's just a snapshot of some of the studies that have been conducted and some of the different effects in terms of biologic markers that have been studied. And a summary of this table on this slide is that MPA affects multiple immune markers, and that NET often doesn't affect the same markers, so leading to potential differences, but also few data points sometimes. You can go to the next slide, please. So to summarize the pharmacodynamics data, just a couple of bullet points. The first is that although peak serum levels of MPA are greater than NET, the trajectories of serum levels of MPA and NET are similar following injection. And then the second bullet is that there are differences in the affinity for MPA and the lack of affinity for NET to bind to the glucocorticoid receptor, and that this affinity of MPA is unique and may lie at the interface of differences observed in the regulation of certain cytokines and chemokines. So that's a summary of the pharmacodynamics data. You can go on to the next slide. I think we want to be at slide 28. So on slide 28, giving an overview of the data that we have that were presented on subcutaneous DMPA. The first is that the availability is rising. It's available in 15 countries and with regulatory approval in 25. That's as of August 2017. And the studies to date primarily establish contraceptive efficacy and the side effect profile, and that is similar to DMPA-IM. And although it's a lower dose than intramuscular DMPA, it maintains a threshold of serum MPA that's sufficient to suppress ovulation for the same duration as intramuscular DMPA. And then the last point is that there are no studies to date assessing HIV risk or the effect of sub-Q DMPA to elicit changes in tissue permeability, target cell recruitment, quantities of immune markers, or microbiome. So many of the biologic markers that have been studied for DMPA-IM and NET have not yet been studied with subcutaneous. So you can go on to the next slide. The next slide, this slide summarizes some gaps in research that were presented during the August meeting and that have also become quite evident as I've been combing through this literature and these presentations. The first is that we have no tissue studies from women using these three methods, and these would be needed to examine differences in tissue permeability or target cell recruitment. Second, there have been no randomized comparisons of MPA and NET with regards to HIV acquisition or pharmacokinetics or pharmacodynamics. Third is that there are not definitive conclusions about the role of any of these injectables in modulating the microbiome. Fourth is that there are not definitive conclusions about the role of these injectables in modulating the recruitment of target cells and which immune markers may lie between contraceptive use or contraceptive initiation and target cell recruitment. And then last, there are very little data available about subcutaneous DMPA and with regards to how it might differ from intramuscular DMPA with respect to HIV risk or biologic mechanisms that could render women more susceptible to HIV. So the next slide, please. This just presents the overall summary, which is very much similar to the executive summary that I first presented. But basically, differences between DMPA and NET-N are prevalent with regards to the epidemiologic associations. Second, there are important differences in the composition of each progestin, MPA and NET. Third, there may be differences in the way that each progestin interacts with potential biologic mechanisms and actual HIV susceptibility. And finally, that DMPA sub-Q follows a similar PK trajectory to IM, but there are few data assessing HIV risk with sub-Q DMPA, and the biological mechanisms that may be elicited there are also a few data about the biological mechanisms that may be elicited to increase a woman's susceptibility to HIV. So you can go, this is my last slide. You've got my email address, but I think that we also have time now for discussion, and I'm really happy to receive your comments and your thoughts and ways to continue working on this report.
These are the people that you might ask whether they want to solve.
Back over to you,
Thank
James.
you very much, Renee, for that presentation. And maybe we'll also hear from those maybe who would have interventions. Please lift up your hand so that you can be unmuted. And in the meantime, I would like to apologize to Sharon Achilles, who we reported was missing from the meeting, but we have just noticed that she's in the meeting. So apologies for that kind of misstatement. So I can't see anybody who has raised up their hand. I would like to maybe propose first that maybe Saad, you'd like to say something about the work you are planning in Tanzania regarding communication.
Yeah, sure. And thank you, James. Thank you, Petros, and thank you, Rennie. Yeah, currently we had approval for the protocol in Tanzania to pilot test the HC-HIV framework, communication framework. And currently my colleagues are in Tanzania training the health providers on the counseling process. And I think very soon we'll start the implementation process of the research, and we'll keep you updated as soon as we start to get results from the field.
Okay, thank you very much, Saad. I think, Kim, you had your hand up.
Hi, yeah, I think you've covered our activities fairly well in your conversation. So we just look forward to hearing everyone else's efforts in thinking about these issues. Appreciate it.
Okay, thank you very much. And maybe to hear from Justus, any updates on the trial, or if you have any other updates from South Africa?
James, who did you call on? Laneta from
Justus
is on.
Okay, so we can listen to Laneta first. Then we can go to Justus of Mayor.
James, mine was a more specific question. So if Justus has an update, why don't you let him go first?
Okay, thank you, James. We will have protocols to implement 2 trials. The first is a small pilot study. We call it the Witch Injection Part 1 study. And it's a small study with 520 participants. Together, our unit and Jenny Smith's unit in KwaZulu-Natal and Janet Hapgood at the University of Cape Town, a randomized clinical trial to compare various immune, biological, physiological, hormonal, and psychological differences between DMPA-IM and NET-N. We have put in a grant application to the South African Medical Research Council, and we expect to hear at the end of the month whether or not that is successful. And if it is, we hope to be able to start soon with that trial. But that's a fairly small trial with a duration of 7 months. The more important one is the main Witch injection study, which is a planned clinical randomized trial comparing HIV risk and other outcomes between DMPA IM and NET-N. And for that, we've applied to funding to the UK MRC Joint Global Trials Grant. We've been informed that we've passed through the first stage, so our summary proposal has passed the first stage, and we're now busy preparing a final proposal, and we'll learn the result of that application in the middle of the year, June, July of 2018.
Yeah, thank you very much, Justus, for that update. So, Lanetta, you can go ahead.
I just wanted to thank Renee for synthesizing the complex information that was discussed in our August meeting, but I had a specific question and comment with regard to the conclusion about, I think it's a terminology thing, a similar PK trajectory for DMPA IM versus sub-Q. And I think that it would be good to revisit that language. I know we had a lot of discussion. There wasn't real necessarily consensus around it, but that it does appear to be at a minimum in the first month different Cmaxes and different exposure. And so the way it's currently phrased, to me, when I read that, implies that they're the same. And we don't have direct comparative data. That was one of the recommendations out of the meeting. So I guess it was just a plea to consider rephrasing that term, a similar PK trajectory, to reflect potential differences, particularly in the first month of exposure.
Thanks, Lenita. I think we can absolutely do that, and I'll go back and make sure that I'll go back and check and make a change, especially to make it sound so definitive, and review the data as well, and review the slides, and I can talk more with Petrus about the discussion.
Perfect.
Thanks for bringing that up.
Thanks, Renee.
Yeah. Thanks, Lanita. I think my other question may be also to you. At the meeting, there was discussions about studies being done on to actually compare DMPA IM and sub-Q PK head-to-head. Do you know whether they've been? Does anybody have any update whether there are any such studies that are planned soon or happening?
I can just say that we had planned in relation to several of our ongoing studies to include an IM group, and we had thought that we were close to getting funding, and then that situation changed. So we're back to approaching additional donors to include. This would just be PK because it would be added into our ongoing PK studies of Depo sub-Q. So those are, as I said, we've been in discussions with several donors that don't have secured funding. Okay. That is not, our proposal did not have to do with what we discussed and what Sharon presented around mechanistic studies. It's purely PK because it's being added into other purely PK studies.
Okay, thank you. So Sharon?
Hi, yes, I wanted to echo what Lenita was saying with respect to revisiting the language of a similar PK trajectory. And I also wanted to note that in the recently published commentary by Chelsea, me, Janet, and Zednek, we review those data pretty carefully, and we make some comments about the PK trajectories, also noting that there are no direct head-to-head comparisons, so that we don't actually know with respect to head-to-head comparisons. With non-head-to-head comparative data that are available, it appears probable that there may be differences in PK within the first 30 days, as Lenita was also alluding to.
That's perfect. Thank you, Sharon. I'll go back and read that section of their paper again. And it's sounding to me like I also should add make sure that there's a research gap noting this, that the lack of head-to-head comparisons and the specificness of that first month after injection. Does that
think
sound
that would
right?
be great. Yeah, that's perfect.
Okay, great. Thank you.
Maybe.
Yeah, thank you, Sharon. And I think that really just points to the urgent need of having these head-to-head studies comparing the PK of DMPA IM and DMPA sub-Q. I think there would be very important studies to do at this time and would be very informative in our decisions as we move forward. I don't see any other hands raised, so I'll just go ahead and maybe highlight some of what we see as the next steps for us here in WHO. And I think one of the first things that we are going to do is to update the status of the follow-up actions based on what we have had, and maybe on a further follow-up with colleagues to hear what I think some of the actions are still in progress, and we'll keep a close watch on what the outcomes are. So that is one of the critical, important next steps. Then now we have a draft technical report from Renee, which I think she's going to finalize based on the input that has been received in this webinar. And any other persons who may have input from the presentations, please do send an email either to us or to Renee at the email that she gave. And that technical report, we are then going to work on it. There were proposed people who were at the meeting who are proposed to kind of develop this package or this review. So based on the report that Renee will develop, then we will be bringing together the people that were proposed at the meeting to maybe work in the next 2 months or so to develop a detailed draft technical report that we'll be able to share with all the participants. And then based on this sharing of this report, then we will probably have a follow-up discussion on how to proceed further, and also as we are all continuing to eagerly await the echo results and to plan various scenarios of what we will be our actions depending on the various results. So I think those are the 4 main areas that we'll be following up on. I don't know whether maybe I've missed some issue or something which anybody would like to add. I don't know, Jennifer, you have your hand up or you had a question?
Yeah, hi, James, and everyone. Hi, thanks. Sorry, I'm a couple minutes after we were having discussion. Thank you to James and Petrus and Renee for those great presentations. This was very helpful for us to be a part of this discussion. I was just curious to hear if any of you could expand a little bit on what specifically is happening to strengthen the national actions that are being taken in response to the updated MEC in countries like Kenya, Uganda, South Africa.
Yes, so Jennifer, there's a lot of activities that WHO has been doing in a follow-up to a meeting that we had in Johannesburg, I think in December, on 10th of last year. And we invited the high prevalence countries to that meeting, basically to brief them on the updated MEC guidance and to start planning country actions. There were 5 countries that were identified as a priority for follow-up, and we are actually having a follow-up webinar tomorrow where these countries will be participating and reporting on the actions that have been taken in country with the support of WHO to have and so the WHO has worked with the countries to try and update national guidelines and take specific actions around updating the approaches in terms of contraceptive delivery programs. We haven't, I don't want to preempt what the countries will report, but at least from what I gather is that in all the countries there have been some activities. The other issue I would like, and I think Petrus presented on his presentation, is that on 15th of February, we are going to be launching the Global Handbook for Family Planning Providers, and it has very specific proposed messages. Of course, as WHO, we cannot be prescriptive to countries. It's a decision of the countries, but we have at least proposed specific ways in which counseling around this issue can be done. And I think also Saad presented from USAID that they are having kind of an implementation research or a learning process which they are implementing in Tanzania to use the framework that was developed as a strategy for communicating DMPA and HIV infection risk. So there are quite some activities that are going on. I don't know, Petrus, maybe you have something to add? Or
thanks, James. I think you've summarized that very well. Laneta, that as James mentioned, this is a dissemination meeting that we had in April last year in Johannesburg in the 10 most affected countries of HIV, where we specifically had the Ministry of Health and WHO officers together, and then USAID and some other people were also there, and we will have a follow-up meeting for that. So this is really nothing to do with the disaggregation of DMPA, DMPA sub-Q and NET-N. This is about medical eligibility criteria that have changed for the injectables. Thanks.
Thank you. Thank you, Petrus. I don't see any other hands up. So I think we can end 5 minutes earlier, which is always a good thing. And I thank you all for participating in this webinar. And I look forward to continuing to work together with all of you on this very important issue. So thank you very much. Have a good afternoon, good evening, or good morning, depending on the time and place. And bye-bye.